Hello students, I am Deepika Bairagee, Assistant Professor, Acropolis Institute of Pharmaceutical Education and Research. So today we will discuss about paper chromatography. So before discussing paper chromatography, we will talk about chromatography. So as you all know that chromatography is an separation technique. It is used to separate the component of a mixture using a fixed, that is called as a stationary phase, and moving, that is called as a mobile phase. It means for separation we required two phases, that is one is the stationary phase and another one is the mobile phase. So basically chromatographic term is come from the Greek word chrome, which is called as the color, and graph, which indicate to write. So in chromatogram, as you can see that we will achieved colored spots. So this indicate that. So recently the IUPAC has defined chromatography as a method used primarily for the separation of the component of a sample, in which the component is distributed between the two phases. As I have already discussed, one phase is the stationary phase while another phase is the mobile phase. So the stationary phase may either be solid or the liquid, supported on a solid or a gel, and may be packed in a column, spread as a layer, or distributed as a film. So the mobile phase may be gaseous or liquid. Now let's talk about the paper chromatography. So paper chromatography was introduced by Martin and Singe in late 1940s in England. Paper chromatography is basically used in almost all the area to solve the complicated problems in chemistry, biology and biochemistry. Now let's talk about the advantages of the paper chromatography. So basically the equipment which is required for paper chromatography is very simple. It is easily available in your laboratories, and it has a high efficacy for the separation, and the separation can be affected on macro, micro and semi-micro scale. So closely related compounds like the homologue, isotopes, isomers and very liable and reactive substance also can be satisfactorily separated by this technique. Now let's talk about the theoretical principle of paper chromatography. So the principle of paper chromatography is based on partition. So it means that the analyte mixture is partitioned between the two invisible liquids, in which one liquid is called as a stationary phase and another one is the mobile phase. So paper basically we use for paper chromatography is made up of the cellulose, which consists of an hydroxyl group. So the OH group in the cellulose are responsible for the hydrogen bonding of the water, and the water molecules hand on the cellulose fiber of the paper act as a stationary phase. Thus this technique is called as a liquid chromatography. As you can see in the structure, there are different types of hydroxyl groups are attached together. Now let's talk about the RF value and the variation. So RF value can be defined as the distance traveled by the solute upon the distance traveled by the solvent fund, in which R indicates the fraction of the solid molecule in the solvent at any specified time, and RF is a function of the partition coefficient, and it is a constant for a given substance provided the condition of chromatographic systems are kept constant. So for greater reliability, we can use reference standard in parallel run of, or you can use the internal standard. As you can see in the diagram that two components are seen, A and B. So A is your sample and B is the reference standard, and you can see that here it is a start line where we can put our sample, and you can see another line of the solvent front from where we can run the solvent. Then sometimes the solvent funds turn the paper, then the position of the individual spot is measured related to the position of the standard substance, that is denoted by the X. So the fraction of the solid molecules can be defined as Rx equals to distance traveled by the substance upon the distance traveled by the standard substance. Now let's talk about the types of paper chromatography. So basically there are two types of paper chromatography. First one is the paper partition chromatography, and second one is the paper adsorption chromatography. So the paper partition chromatography is utilized as a support with one solvent as a mobile phase and other as a stationary phase. Here we can use both the phases as a liquid. So the migration of the substance is due to the difference in their partition coefficient. Then in paper adsorption chromatography, one phase is the solid and another phase is the liquid. So in some cases the paper to be used is coated or impregnated with the adsorbents like the silica, gel or alumina. So the modified paper is used as an adsorbent and the solvent is allowed to flow over the unknown component. So the migration of the substance is due to the difference in their adsorptive power of the substance to be separated. Now let's talk about the operational technique. So for performing the paper chromatography will require these steps. First of all, you have to choose the filter paper, then you can use the modified filter paper, then how you can prepare the paper, it is also we will discuss, then how you will prepare the sample, then how you will apply the sample, then what kind of solvent we can use for performing the paper chromatography, then the chromatographic tank or the chamber, okay, what kind of the chambers or tanks you can use. Then in the next step we will learn about the development of the chromatogram, then and in the next step we will see the drawing of the chromatogram and how we will spot the location of that particular spots. So first of all, we'll talk about the choice of the filter paper. So specially manufactured filter papers can be used for paper chromatography. So particularly we will use Whatman filter paper. Then this filter paper contains 98 to 99% of alpha cellulose and 0.07 to 0.01% of the particular minerals we can use. Then it also consists of the beta cellulose, ethyl soluble matter, ammonia and the lyophilic substance are also present. Then the dimension of the paper should be 46 to 57 and 58 to 68 centimeter, and each peg contain 100 to 500 sheet into it. So you can cut that chromatographical paper according to your size, whatever the size you require. So basically we will cut it into the rectangular or square paper. Then the speed of the chromatographic analysis is depend upon what kind of Whatman filter paper you are choosing. So the coarser and faster paper are used basically for the development of the chromatogram. So Whatman filter paper 31, there are different numbers of Whatman filter paper. So 31 preferred over the Whatman paper number 1 because it is coarser. So now let's talk about the modified filter paper. So for efficient separation of certain substances, especially treated or modified filter papers are used, that is buffered or treated paper like Whatman phosphate, Whatman citrate or paper treated with alumina or silica acids are used. So in case of reverse phase chromatography, paper is impregnated in the mobile phase, and that is the non-polar solvent we can use. Then afterwards we will dry it and then used. So example of chromatographic paper, we can use different kinds of chromatographic papers. So generally we will use Whatman filter paper, and if we want to perform the ion exchange chromatography, then ion exchange papers like D81, P81, strong cation paper, SG81, non-charged silica gel cellulose paper. These are the examples we can use. Then let's talk about the preparation of the paper, that how you will prepare the paper. So once the paper is decided that which type of chromatographic technique you want to perform, so according to that we can use that particular um, filter paper or what kind of the analyte you want to separate. So according to that we can choose the paper. So you can cut it into the desired size and shape depending upon the work to be carried out. So generally rectangular and square shaped are used. So after noting the direction of the run on the paper, start line is marked. Then how you will prepare the sample. So the mixture to be separated is applied to the paper as a solution. So there is no particular procedure given. So you can prepare that sample, and 10 to 20 microliters you can apply it on that filter paper. Now application of the sample. So the sample may be applied as a spot or in the form of band you can apply. So generally the size of the spot should be small as possible. So diameter of the sample not exceed 5 mm. And then the quantity of the sample applied to the paper is important rather than that of the volume. So that in what concentration you are applying that particular sample, it is important. The micro syringe is used for the application of the sample, so that the proper size of the sample can be applied onto the paper. And for quantitative purpose you can use capillary for applying that particular spot. A narrow strip like 5 to 10 mm long and 25 mm apart, or a single spot at least 10 mm apart should be applied on that paper. Then what kind of solvent we can use? So the selection of the proper solvents depend upon the nature of the substance to be separated. What kind of the analyte you want to separate, it depend upon the polarity. So factors which affect this selections are viscosity, surface tension and polarity of the solvent. So the solvent used in the paper chromatography are given in the increasing order of the polarity, like N-hexane, cyclohexane, carbon tetrachloride, benzene, toluene, trichloroethylene, diethyl ether, chloroform, ethyl acetate, and butanol, and propanol, acetone, ethanol, methanol, water. So if pure solvent is not satisfactory for the separation of your component, then you can use the mixture of the solvents. Now let's talk about the chromatographic tank or the chamber. So basically glass, plastic and stainless steel chromatographic tanks are used, and glass tanks are preferred or most commonly used, as in this chromatographic technique we can see how your chromatogram is developed by using the tank of glass. So they are available in different sizes depending upon the length or the breadth of the paper, and type of the development. So the chambers or the tanks have lid with a hole for introducing the solvent through it. Now let's talk about the development of the chromatogram. So sufficient amount of the solvent should be present in the chamber. So during the development, paper should be freely suspended and should be vertical. Large temperature changes and exposure of the light should be avoided, so that the separation should be optimum. So the atmosphere of the chamber should be saturated with the solvent paper, and the paper dipped into the solvent, that the spot will not dip completely into the solvent. You have to carefully observe that your spot is not dipped into the solvent phase. Now, drying of the chromatogram. So the wet chromatogram after development is dried in a special cabinet heated electrically with the temperature control. You can use dry oven, and for drying can also be carried out by transferring the chromatogram into the racks and putting into the drying cabinet. So they are dried by cold or hot air depending on the volatility of the solvent, that how much your compound is volatile. Then let's talk about the location of the spot. So once the chromatogram is developed and dried, so next step is to locate the spot. So if the substance are colored, they are visually detected, but for colorless substance, various methods can be used, like the physical method, in which you can use the UV light for the detection of the fluorescence and radioisotope component, and the chemical methods like the spraying reagent, the glass atomisers are used. Spraying reagents like the ninhydrin solutions you can use. Basically chromatographic technique is a qualitative or quantitative technique. So for the qualitative analysis, the simple measurement of the RF value either by the comparison with the reference substance or by the standard value in the literature is done. You can use for the qualitative analysis of your sample, and for the quantitative purpose, you can use basically by visual comparison of your spot and by measuring the area of the spot. So visual comparison of the spot is several reference solutions containing different concentrations are needed, and the spot is compared for its size and intensity of the color by fluorescence or UV observance. So the measurement of the area of the spot, when the outline of the spot or the zone are well defined, the size of the spot may serve for determining the quantity of the substance. So the area can be measured with the planimeter or graph paper. So you can use planimeter or graph paper for the measurement of your spot. Then radio tracer analysis. So the radioactive element which is labeled is used to locate or determine the quality of the material on the chromatogram, or the compound of the chromatogram is identified by subjecting to neutron bombardment. So the activity and the locations are measured by passing the paper either in the gas flow containing the chamber or a thin window by Geiger-Müller tube. Now next method is removal of the substance from the paper. You can remove that particular substance from the paper, that particular spot from the paper, by the elution method. So for the quantitative analysis, the substance is removed by elution from the paper. So the simplest procedure is to cut out that particular part of the filter paper and soak into an optimal amount of the solvent. So the extraction may be done by shaking or by warming. So the elements are obtained from the chromatogram, may be diluted or concentrated and then analyzed by any suitable technique. Now let's talk about the development techniques. So there are different development techniques in paper chromatography. First one is the descending chromatography. As you can see in the diagram that the solvent can be flow in descending manner. So the apparatus consists of a well-sealed glass tank of a suitable size and shape provided with a trough for the mobile phase in the upper portion. So we will run the mobile phase from the upper portion in the descending manner. So the paper with the sample spotted is inserted in the trough containing the mobile phase. The jar itself having been equilibrated with the mobile phase prior to the illusion. Since the movement of the mobile phase is descending, so it is referred as a descending chromatography. Basic advantage of this chromatography can be continued even though the solvent run off at the other end of the paper. Now let's talk about the ascending chromatography. So the solvent stream moves upward. The mobile phase is placed in a suitable container at the bottom of the chamber or in the chamber itself. So the sample are applied a few centimeter from the bottom edge of the paper suspended from the hook, and the paper may be rolled into a cylinder held together by a straps or the plastic clip. So in the ascending manner, the solvent can be moved. Now let's talk about the ascending descending chromatography. So this can be performed in both the way, ascending and descending. So as you can see in the diagram, the hybrid of the above two technique is called as ascending and descending chromatography. Here the upper part of the ascending chromatograph can be folded over a glass to change over into the descending after closing the glass rod. Now let's talk about the radial or disc chromatography. In radial chromatography, we can cut that particular filter paper into circular portion, and from the radius or edge of that particular center, we can cut the wick, and the sample is deposited at the center of that particular paper at the upper end of the wick, and the paper is then led into the edge of the circular disc with the wick dipping into the solvent at the bottom of the dish, and the liquid ascend by the wick and flow radially through the paper. Now next technique is multiple chromatogram. So in this technique wherein the irrigation is repeated either in the same direction as in the first run or in the direction perpendicular to the first system. So multiple time we will run the sample into the solvent. Now next technique is two-dimensional chromatography. In this we can run the chromatogram into two directions. So the paper is cut into square or rectangular, and the sample is applied to the one corner of that particular paper. So using a solvent system, first development is carried out as per the ascending method, then the paper is taken out and dried, and the second development is performed at the right angle of to the direction of that particular first run. And then the larger the area of the paper, more complete the resolution of the complex mixture can be achieved. Now let's talk about the application of the paper chromatography. So basically it is an inexpensive technique and rapid method that provide reliable results. So it is used both as a qualitative and quantitative identification of your particular analyte, and it is widely used for the separation of amino acids. It has been proven in the analysis of the substance including protein, peptides, amino acids, poly, oligo, di and monosaccharides, natural products, steroids, bile acids, pigments, dyes, and inorganic species. Then separated compounds can be recovered by cutting the spot, dissolving in a suitable solvent, and evaporating the solvent. And basically it is used in several scientific studies for the identification of unknown organic and inorganic compound formed from a mixture. Then phenolic substances such as anthocyanin, flavonoids, and other polyphenolic substances can also be separated and identified. In forensic study, also paper chromatography is used in crime scenes investigation and DNA and RNA sequencing along with the other studies. In paper chromatography, it is used as an analytical chemistry technique for identifying and separating of colored mixtures like the pigments. For the separation of sugars, amino acids, lipids, and nucleic acid and other biomolecules can be easily identified by spraying with appropriate reagents to detect this specific compound, or it had been used for the taxonomical and genetic study. Example, taxonomical study in the molluscan genus Laminia, or it is used for the study of structure of the steroids. So that's all. Thank you so much, students.